Okay, let's talk a little bit about the activation and the differentiation of T cells. So, for a naive T cell, again, naive meaning that it has not encountered its specific antigen, in order for the naive T cell to become activated, a two-step process must occur. So, we have antigen binding and then we have co-stimulation. So the first step is antigen binding, and this is where the T cell receptor, called a TCR, is going to bind to the antigen MHC complex on the surface of the antigen presenting cell. The TCR has to undergo what we call double recognition, meaning that it must recognize both the antigen and the MHC molecule presenting it. The second step is going to be co-stimulation, where the T cell must also receive additional signaling molecules called co-stimulatory signals from the antigen presenting cell. Without co-stimulation, this T cell is going to enter into a state of anergy where it becomes unresponsive and it cannot divide or secrete any cytokines. So this is a really important safety mechanism to help prevent autoimmune responses. If both of the signals are present, the T cell is going to become fully activated. It's going to enlarge. It's going to undergo clonal expansion, meaning that it's going to rapidly divide to form many identical effector and memory T cells. So this is just showing you that process that we were just talking about once we have activation. So we can first see the antigen presentation by the APC. We also have double recognition and then our clonal expansion. So the differentiation of those activated T cells are guided by cytokines, and cytokines are just small signaling proteins, and they're secreted by a variety of different types of immune cells. So IL-2, or interleukin-2, is the key growth factor. It's especially important because it promotes rapid T cell proliferation and further activation. We also have some other cytokines, one of which we talked about previously, that was interferon gamma, and this was going to help boost macrophage activity and amplify both the innate and adaptive immune responses. So the primary T cell response usually peaks within 7 days, and then once that threat is eliminated, most of those activated T cells are going to undergo apoptosis, that programs cell death, between days 7 and 30. And that's going to help prevent chronic inflammation and potential tissue damage. However, remember, we have that pool of memory T cells that's going to remain in our body to help provide a faster and stronger secondary response in the event that we encounter that same antigen again. So the cellular immune response is really, really specific, and it is tightly regulated type of defense mechanism that's going to rely on our T cells to help identify and eliminate any infected or abnormal cells. And again, it's going to involve the antigen recognition through the MHC presentation, careful activation through a 2-signal system, and also coordination of immune activities using those cytokines that I just mentioned.